Okay, I'll introduce our next speaker, Bin Lu. It's going to be our final talk for the session. So please, Bin Lu from the Institute of Psychology, Beijing.
Okay, hello. Oh, sorry. Hello, everyone. I'm honored to introduce our recent work about an MRI-based interpretable deep learning model for Alzheimer's disease. I will try to keep it brief to save your dinner time. Sorry, Xiaoyu, we use the same picture because it's a really good illustration of Alzheimer's disease. And we all know that Alzheimer's disease is one of the most prevalent neurodegenerative disorder in the world. And the diagnosis is mostly based on the clinical guidelines. And currently, brain MRI hasn't been integrated as a diagnostic biomarker. And mild cognitive impairment can be an early stage of Alzheimer's disease. And more than 50% of them would progress to Alzheimer's disease in 5 years. So luckily, we have several anti-amyloid treatment these days. And it's also increased the need for early intervention and early detection. And the current study is basically an extension of our former work, in which we build a AD classifier based on transfer learning approach. We applied almost all the open access data set in the world and got more than 85,000 T1 images from more than 200 sites. And we used the DeepRB to pre-processing them and got gray matter density and volume maps to train a convolutional neural network. And the convolutional neural network is for sex classification, and it achieved around 95% accuracy in cross dataset validation. After that, we fine-tuned the model to an AD classification model. Finally, the AD classification model achieved over 96% AUC and over 90% accuracy in 4 independent datasets, showing good generalizability. And the model also shows some potential to predict the progression of MSI participants. I mean, the MSI converters are 3 times more likely to be predicted as AD than the non-converters. And we also use the occlusion map to find the most important brain area for model to identify AD. And they are, of course, hippocampus or parahippocampal gyros. And that brings us to the current study. We're trying to further validate the model in AD screening and progression prediction in a Chinese dataset called SEALCode. And we want to further move from group-level occlusion map to individualized brain risk maps. And we want to also evaluate the correlation between the brain MRI-based scores and the cognitive and the plasma biomarkers. Here are the results. The model achieved an accuracy of 88% and an AUC of 91% in the SEALCode dataset. Considering the age and ethnic distinct between the SEALCode dataset and the original training datasets, these results are quite encouraging. As for progression prediction, we can find that over the 60 participants who eventually convert to AD, the model identified 52 of them at baseline, reaching a capture rate of about 86. And if we do not just use the baseline data, but also use the whole longitudinal data, we can find maybe over 60% of the MSI participants who eventually progress to AD can be identified 2 or 3 years before their first diagnosis. It kind of maybe open a potential window for early detection or even intervention. We can also see the results from survival analysis. And the red lines indicate the high-risk groups and the blue lines indicate the low-risk groups. And we can find that either in the full sample or in the MSI sample, the high-risk groups show a significant faster progression rate compared to those in the low-risk groups. And next, we find that the brain MRI-based risk scores are correlated with most plasma biomarkers and cognitive measures across the whole data sets. It's not that surprising because we know that it may be driven by the between group differences among the groups. I think what's more important is that if we look into each group, we can find that the plasma biomarkers such as p-tau217 and NFL are correlated with the brain-based scores in healthy control group. But for the cognitive measures like MMSE, the overall cognitive measures, they are correlated with the brain MRI-based scores in MCI groups. It kind of fits the notion that the changes in the plasma biomarkers tend to appear earlier than these changes in the cognitive measures. Next, we want to generate individualized AD brain risk maps. To do this, we first created an average brain template. And the predict score of this template is exactly 0.5, just right between the most severe AD and the most healthy control. And if we got a new participant, we can replace the template with the actual participant's data for certain brain area. And we can treat the predicted score of the combined image as the disease contribution of this brain area. And if we do this region by region, we can finally got a whole brain risk map for single subject. But we have to say that the identified high-risk brain areas is just based on the structural MRI and under the current model setting. Well, they might be sensitive, but they lack specificity and should be interpreted with caution. Here we want to illustrate a potential use for these brain risk maps. We try to cluster all the AD risk maps into 3 subtypes, and we stratified all the MSI participants into these 3 subtypes. And what's interesting is that we find, although the 3 subgroups of MCI didn't show any significant differences between in baseline cognitive scores like MMSE, but the subgroup 3, they showed significant faster cognitive decline than the other groups. I think it suggested that we can use these kind of risk maps to identify MSI participants who are more likely to benefit from closer monitoring or even early intervention. To summarize, this brain MRI-based model generalized well to an age and ethnical distinct sample. And it showed meaningful correlation with cognitive and plasma biomarkers and generate individualized interpretable risk maps with some potential use. You can also try it yourself. You can either use the DeepAbbey built-in tool or try our online demo dataset. Although the design of this website is kind of in the early 2000s one, but it actually works. I'd like to say thank you to all my colleagues in this project. They are Chen Yanrong, Li Ruixian, and Zhang Mingkai. And I'd like to also say thank you to all my supervisors in this project. They are Yan Chaogan, Han Ying Francisco Javier Castellanos, Paul Thompson, and Christian Beckman. Thank you for your attention.
Thank you, Ben. That's great work. Questions?
That was a really nice talk. Thanks a lot. So this is kind of almost following up a question that Nathan asked from the previous talk. So obviously there is some sort of subcortical involvement in a lot of these diseases, and I was interested to see in your kind of last subtype spatial map, it seemed like there were parts of like the midbrain that were contributing at least to some of these subtypes. And this is something that we really haven't been able to image very well with neuroimaging, and we don't really have good MRI markers for it. I'm just wondering, are you finding that particular part of the brain, like the midbrain, is actually something that's contributing to your predictive model? And if so, do you think this is actually measuring some sort of neuroanatomy, or do you think this is something more of like technical with the MRI or something like that?
Personally speaking, I think both is possible. Maybe we should deeper the research in like using PET or the other technologies, but only use MRI, and I don't think I can like give a certain answer to this question. Yes, it's quite surprising to us too, because the subcortical regions for like Alzheimer's disease is kind of not that ignored to the nowadays research to some extent. Thank you.
Yeah, thanks.
Another question?
Hiya, super interesting, super great talk. I was just wondering, again, on this last slides, it really struck me the different subtypes of AD that you were showing, and you looked at cognitive profiles. I was wondering if there's any difference with like disease duration. So how long do you see a difference in the different subtypes based on whether they've had a diagnosis for, over time?
Yes, it's a really good question. Yes, we should have checked this duration. Yes, but we didn't. But the age, they did show the significant difference between of the age between the groups, and but the duration, I think, is another thing. We should check it.
And just to follow up on that, do you have measures of disease, like functional disease severity, available? Beyond the cognition, actually, how they're...
Yes, unfortunately, we don't have that much. The sample size for the people with these cognitive measures, like the specific measures, is not that much. So they didn't show any significant results. But yes, thank you.
Great talk, well done.
Thank you.
Okay, thank you. I have one question on the app here, but I think it might be one that's better asked in person because it's a little vague. So is the person who posted a question in the room now? I assume they might be. No? Okay, all right, I'll ask it. So the question is, could I use Brain ImageNet to predict AD conversion in my data set? So I'm not sure what the data set is specifically, but...
Yes, if you means that you use the online demo data sets or some built-in tools to predict, you should use it with caution because it's just some basic research. You shouldn't actually use it in the clinical setting. And if you want to build your model by your own, you can also use our open access model or the everything. Thank you.
Okay. All right, great. Are there any more questions in the room? No? All right, well, join me in thanking Bin for a wonderful talk. Okay, so we're 10 minutes ahead of our scheduled end time, so please feel free to leave, or if you have any other questions for our panel of speakers, please come up the front, but otherwise, please join me collectively in thanking all of our speakers for the session. And one more personal request, if anybody has a black notebook next to them that doesn't belong to them, please let me know because I've misplaced my notebook. Thank you.